Okay, so welcome back to this next video in which we are discussing heterotrimeric G proteins and their effects on adenylyl cyclases. Okay, so we're just in the process of discussing the classification of adenylyl cyclases, okay, and we've seen that adenylyl cyclase enzymes are going to be classified into these four groups. Group 1 is the calcium-activated adenylyl cyclases and contains adenylyl cyclase 1, 3, and 8. Group 2 is the G-beta-gamma stimulated adenylyl cyclases and contains adenylyl cyclase 2, 4, and 7. And group 3 is the G-alpha-I / calcium inhibited family of adenylyl cyclases and contains the two adenylyl cyclases, adenylyl cyclase 5 and adenylyl cyclase 6. Okay, so let's highlight these ones. So I have group 3 in vivid purple here, and we've got adenylyl cyclase 5 and adenylyl cyclase 6. And finally, group 4 is the forskolin insensitive family of adenylyl cyclases, and this only contains one member, which is the final member, which hasn't so far been classified. Okay, so this is adenylyl cyclase 9. Okay, so I'll highlight this one in vivid green. Okay, so basically, forskolin is a drug which promotes the activation of adenylyl cyclases. Now, I don't believe it's actually used at all clinically. It's an experimental drug. It's used in experimental science if you want to activate adenylyl cyclases. And basically, it triggers adenylyl cyclases to go from the inactive state where, remember, the C1a and C2a domains are not dimerized, to the active state where the C1a domain and the C2a domain are dimerized. And in that state, you have an active enzyme which is capable of converting adenosine triphosphate molecules into cyclic adenosine monophosphate molecules and pyrophosphate molecules. Okay, but the only adenylyl cyclase which forskolin doesn't work on is this adenylyl cyclase 9 enzyme, which is why it's put into this category on its own. Right, so we've now classified our 9 separate adenylyl cyclases into these separate families. Okay, now we're going to move off adenylyl cyclases for now and turn our attention to G protein coupled receptors. Okay, and we'll revise GPCRs, we'll revise the heterotrimeric G proteins and things like that, so that we can then put it all together and look at how the heterotrimeric G protein subunits are going to regulate the activity of the adenylyl cyclases. Okay, so G protein coupled receptors, or GPCRs for short. Now, G protein coupled receptors also have another, a number of other names. They're also known as 7-transmembrane receptors, okay? Often just abbreviated to 7TMs for 7-transmembrane receptors. Okay, and GPCR is the usual abbreviation for G protein coupled receptors. Right, okay, so the characteristic feature that all G protein coupled receptors have is that they have these 7 membrane-spanning alpha helices. Okay, so the amino terminus is on the extracellular aspect of the membrane, and then you have these 7 membrane-spanning alpha helices like so, which leaves the carboxylic acid terminus on the intracellular aspect of the plasma membrane. Now, G protein coupled receptors is the biggest family of proteins in humans, basically. There are over 800 known G protein coupled receptors, so these are easily the most common form of receptor that we have in the human body. Okay, and it's not really understood why nature found this 7 membrane-spanning alpha helices structure so useful, basically, because it seems to have latched onto this structure and run with it, basically. You have 800 of these things, and it's incredibly effective, but we don't understand what the significance of the 7 membrane-spanning alpha helices is. Okay, right. So, basically, G protein coupled receptors are also grouped into 5 families. Okay, so they're also classified to help us gain some sort of way of understanding this, because you've got 800 of these things. How are you going to actually comprehend that? Well, the human mind loves classifying things, so we have 5 separate classifications of G protein coupled receptors. So let's discuss these on the other side. Okay, so basically, the most important family of G protein coupled receptors is the rhodopsin family of G protein coupled receptors. Okay, so I'll start with the rhodopsin family. And basically, this family contains 750 of the known 800 G protein coupled receptors, so it's easily the biggest family. Now, they are quite plain compared to some of the others. They don't have some of the fancy features that the others have. Okay, so they do have the 7 membrane-spanning alpha helices. All G protein coupled receptors have the 7 membrane-spanning alpha helices, but they have a very small amino terminal domain. Okay, and the characteristic feature of the rhodopsin family G protein coupled receptors is that they bind their ligand, whatever it may be, in the transmembrane domain region. Okay, so the ligand binds to amino acid residues which are within the 7 membrane-spanning alpha helices. Okay, so the ligand comes in and will bind to the residues within the 7 membrane-spanning alpha helices, and that is what characterizes all of the 750 members within the rhodopsin family, that the ligand binds to residues within the 7 membrane-spanning alpha helices. Okay, and it will then trigger a conformational change, which then allows the G protein coupled receptor to interact with the heterotrimeric G protein, which we'll come on to later. Okay, so most G protein coupled receptors fall into this category. For instance, the adrenoreceptors all fall into this category. The muscarinic acetylcholine receptors all fall into this category. The rhodopsin receptor, which is a photoreceptor, falls into this category. So there are many important examples within here. You can go on. The histamine receptors, the dopamine receptors, etc. They all fall into this category. Okay, right. So next up, we'll talk about a smaller family, but still important, which is the secretin family of G protein coupled receptors. Okay, so the secretin family of G protein coupled receptors has a slightly larger amino terminal domain than the rhodopsin family of G protein coupled receptors. Okay, so let me show these now. So let's say the amino terminal domain looks like so. So here's the amino terminus, and then maybe it has a little bit of a more elaborate structure here before then going into the 7 membrane-spanning alpha helices, like so, and then finishing with the carboxylic acid group on the cytoplasmic aspect. Okay, and basically, the secretin family of G protein coupled receptors include receptors for things like calcitonin, okay, and parathyroid hormone, okay, so PTH. And also glucagon. The glucagon receptor is a very important example of a secretin family G protein coupled receptor. Okay, and what you will notice about all of these, calcitonin, parathyroid hormone, glucagon, is that they are all peptides. Okay, these are not small molecules. We're not talking about serotonin or adrenaline anymore. These are peptides, all of them. So, secretin family G protein coupled receptors generally have a ligand which is a peptide. Okay, and where does this ligand bind? Well, basically it binds in this space between the amino terminal domain that is on the extracellular side and the 7 membrane-spanning alpha helices which are within the plasma membrane here. Okay, so this is where the ligand binds in the case of secretin family G protein coupled receptors. Okay, so this is our second family complete. Let's go on. Let's now talk about the glutamate family of G protein coupled receptors. And you might be able to guess which receptors are within the glutamate family of G protein coupled receptors. Okay, so this family clearly contains the metabotropic glutamate receptors. I want to stress that it does not contain all of the glutamate receptors. The metabotropic glutamate receptors are pretty tame compared to the rest of the glutamate receptors. Most of the glutamate receptors are ligand-gated ion channels, so for instance the AMPA receptors, the kainate receptors, the NMDA receptors. Those are the main glutamate receptors, and they're all ligand-gated ion channels. They're not G protein coupled receptors. The metabotropic glutamate receptors, so this M here is for metabotropic, the glue is short for glutamate, and then the R is for receptors, represent a very small population of the glutamate receptors. Glutamate is the main excitatory neurotransmitter in the central nervous system. Okay, so it's the main neurotransmitter in the brain, basically, along with GABA, which is the inhibitory one. So it's a very, very important system. These receptors play a much smaller part in the glutamate system than the ligand-gated ion channels, but they're still important, and they are within this family. Okay. Other examples are receptors to GABA, the main inhibitory neurotransmitter within the brain. And again, most GABA receptors, the GABA-A receptors, are all ligand-gated ion channels of the cis-loop ligand-gated ion channel family. Okay, so they have a similar structure to the nicotinic acetylcholine receptor. But there are GABA-B receptors, which are within this glutamate family of G protein coupled receptors. Okay, so let's see what's special about these G protein coupled receptors. Well, basically, again, they have a fancy amino terminal domain. I'll try and draw this like so. Okay, so here's the amino terminal, and then we have the 7 membrane spanning alpha helices, like so, and then the carboxylic acid group right on the end, like so. Now, what do these receptors have that's special? Well, they have this domain up here, which is known as the venous flytrap domain. Okay, now why is this called the venous flytrap domain? Well, because it resembles a venous flytrap. Basically, the ligand, whether it's glutamate or GABA, is going to bind up here. Okay, and what will then happen is those two sides will close around the ligand, just like a venous flytrap, basically. Okay, so that's the characteristic feature of these glutamate family G protein coupled receptors, that they have this venous flytrap-like action when the ligand binds. Okay, so the next family of G protein coupled receptors that I want to talk about is the adhesion family of G protein coupled receptors. Okay, so these are kind of boring, okay? These are, unless you're actually a researcher in these G protein coupled receptors, these are generally the ones that no one knows anything about, okay? So basically they bind to components of the extracellular matrix, and they have very large amino terminal domains, like so, okay? And then they have their 7 membrane spanning alpha helices, like so. Here's the amino terminal here. And then they will bind to components of the extracellular matrix, like so. So I'll put this in orange here. So this represents some component of the extracellular matrix, ECM, for extracellular matrix. Okay, right, so that's the adhesion family of G protein coupled receptors, and now we'll finish this discussion off with the final family, which has the by far silliest name, which is the frizzled / taste 2 receptor. Okay, now these are G protein coupled receptors, and they have ligands, so they have more normal ligands. Well, they have the more typical ligands than adhesion family G protein coupled receptors. And basically they bind their ligand extracellularly, but they don't have a venous flytrap domain. Okay, so they're all grouped together as, they're not rhodopsin family G protein coupled receptors because they don't bind it within the transmembrane domain. They bind it in the amino terminal domain, but they don't have the venous flytrap domain that the glutamate family G protein coupled receptors have. Okay, so here maybe is the ligand, like so. And this family is named after the two most famous members of it, basically, which are the frizzled receptor and the taste2 receptor. Okay, so frizzled is part of the Wnt beta-catenin pathway, and its ligand is Wnt. Okay, now it also has a co-receptor which is with it and which responds to Wnt. The taste2 receptors, those are involved in the gustatory system. Okay, so these are receptors for bitter taste molecules, basically, so they're in the tongue. Okay, right. So that then concludes our discussion of the 5 separate families of G protein coupled receptors. What we now want to talk about is how they interact with heterotrimeric G proteins. Okay, so let's now turn our attention from the G protein coupled receptors to the heterotrimeric G proteins. So let's have our plasma membrane here again, and the above portion will represent the extracellular matrix. The extracellular fluid, which will have the extracellular matrix within, and then this side will represent the cytoplasmic domain. And basically attached to the membrane, but not implanted into it. So these are not integral membrane proteins, but they are attached to the plasma membrane, and I'll show you exactly how. You have heterotrimeric G proteins. Okay, so we'll start with the main member of the heterotrimeric G protein, which is the alpha subunit. Okay, so this represents the alpha subunit of a heterotrimeric G protein. Now highlighted in vivid purple here. And basically, the alpha subunits have palmitic acid molecules stuck onto them, or myristic acid molecules stuck onto them. Okay, some of them even have both. So every alpha subunit, and we will see that there are many different types of alpha subunit, every alpha subunit that you can dream up will actually have a very long chain carboxylic acid molecule stuck onto the side of it. Okay, and this is what this little line here represents. So this will either be myristic acid, which is a 14-carbon fully saturated carboxylic acid, okay, or it will be palmitic acid, which is a 16-carbon fully saturated carboxylic acid. So in the more modern nomenclature, myristic acid will be called tetradecanoic acid, and palmitic acid will be called hexadecanoic acid. Okay, now alpha subunits will at the very least get one of these put on, if not both. Okay, and these will dangle off the side, and what they're brilliant at doing is implanting into the inner leaflet of the phospholipid bilayer. So remember, the phospholipid bilayer consists of 2 layers of phospholipids. The outer layer is then called the outer leaflet, okay, and the inner layer is called the inner leaflet. Okay, so these palmitic and myristic acid groups are going to basically implant into the inner leaflet of the phospholipid bilayer, and that will anchor the alpha subunit at the inner leaflet of the phospholipid bilayer, hence holding it onto the base of the plasma membrane, basically. Okay, right. Now, alpha subunits can be in 2 states. They can either be in an on state or an off state. Okay, now in the on state they have guanosine triphosphate GTP bound to them, and in the off state they have guanosine diphosphate GDP bound to them. Okay, now in the off state, when they have guanosine diphosphate bound to them, the alpha subunit will associate with 2 other subunits, and these other 2 subunits always remain together in a complex. Okay, so these are the beta and the gamma subunits here. Okay, so beta and gamma always remain bound together. Okay, under no physiological circumstances do they split, and therefore they are often collectively referred to as the beta-gamma complex, but it is fundamentally 2 separate proteins. Okay, now Basically, the gamma subunit also has a lipid molecule, a long-chain hydrophobic molecule stuck onto the side of it, okay, which again latches into the inner leaflet of the phospholipid bilayer and holds the beta-gamma complex to the inner leaflet of the phospholipid bilayer. And it's lucky for the beta subunit because it has no such lipid modification. Now, the lipid modification of the gamma subunit is not a myristic acid or a palmitic acid molecule. It's something a little bit more complicated to explain, called a prenyl group. Okay, now if you're interested, if you watch my video on introduction to G protein coupled receptors, actually no, not introduction to G protein coupled receptors, if you watch my video on heterotrimeric G proteins, we go through lipid modification of these subunits completely, and we discuss exactly what a prenyl group is. Okay, I'm not going to go into it here because I take absolutely ages to explain it, so we've got enough to get through, but basically it's a polymer of isoprene molecules, if you know what that is. Okay, right, so let's colour these beta and these gamma subunits in. So the beta subunit we'll have in turquoise here, and then we'll have the gamma subunit in pink as well. Okay, so the beta subunit basically latches onto the gamma subunit, and because the gamma subunit has this prenyl group which is attaching it into the inner leaflet of the phospholipid bilayer, both the beta and the gamma subunit get latched onto the inner leaflet of the phospholipid bilayer. And when the alpha subunit is off and has GDP bound, it will associate with a beta-gamma complex, basically. Okay, so we'll call it there for this video. In the next video, what we want to discuss is the many different alpha subunits, the many different beta subunits, and the many different gamma subunits. Okay, so I'll see you in the next video.